Welcome to everybody today. My name is Emily Sena. I'm a senior lecturer at the University of Edinburgh and am on the board for the BNA's credibility campaign. I want to say an extra special thank you for joining us today, actually. I think particularly at a time where there's lots of ongoing global events that I think will be affecting the neuroscience community as much as many others in lots of different ways. So I think we're particularly grateful for your time and attention this afternoon. So this is one of 3 webinars on in vivo research that the BNA is running as part of our work to strengthen the credibility in neuroscience. The campaign is being supported by the Gatsby Foundation. The format for this afternoon is compact, which I think is helpful when we're trying to keep your attention. We've got the wonderful Esther Pearl joining us. She's going to speak for about 15 minutes, and then the rest of the half an hour that we've got available will be for questions. It would be great if those of you attending could use the Q&A tab for your questions, and then rather than using the chat function, but you can use the chat function obviously for general discussion. I also want to mention that this webinar will be recorded, and will be made available on the BNA's YouTube channel for people who want to share it and go back and look at it later. So like I said, we're very fortunate to have Esther with us today. Esther's a program manager for experimental design at the NC3Rs, which is the UK's National Centre for the Replacement, Refinement and Reduction of Animals. Esther's got a great globe-trotting CV. She completed her PhD in biochemistry in New Zealand at the University of Otago, and then worked with Xenopus as a postdoc in developmental biology in Montreal, but also in London at King's College London, I guess before joining the NC3Rs. So I don't want to take up too much time because, like I say, it's a very bite-sized session we have today. So I'm going to pass on to you, Esther, and I look forward to a great talk and to the great discussion we'll have with audience members afterwards.
Thank you, Emily. I'll just go ahead and share my slides with you guys. Right, so as Emily said, I am Program Manager for Experimental Design at NC3Rs, and I'm going to talk about improving reliability in our research. And first, we'll start with acknowledging that our results are not always as reliable as we'd hope. So you've probably heard of issues with the reproducibility of animal research, and this is a topic that has been discussed a lot recently, and these are just a few of the headlines from the last few years. So some groups have actually tried to assess the extent of reproducibility issues, and the Reproducibility Project Cancer Biology is one of these groups. And so they launched in 2013 with the aim of replicating 193 experiments from 53 high-impact cancer biology papers, and they looked at papers published between 2010 and 2012. And overall, the project found that 59% of experiments that they tried to replicate couldn't be replicated. But what I found particularly interesting is that they really had to reduce the scope of the project. So by the time the project was completed, they performed only 50 experiments from 23 papers. And the main reasons they gave for having to reduce scope were a lack of methodological details in the original papers, and materials such as like mouse lines that have been bred specifically for projects were not available. And the project lead actually had this quote in one of the interviews after the project finished, which I thought was really interesting. He said, "None of the 53 papers selected contained enough detail for researchers to repeat the experiments. So replicators had to contact authors for information such as how many cells were injected, by what route, or the exact reagent used. Often these were details that even the authors could not provide because the information had not been recorded or laboratory members had moved on." So that shows that one of the issues that we have with reproducibility and the reliability of our results is also about how we record those and how we report those. So let's have a look at some of the other causes of reliability issues. And so they're really well highlighted in this report from the Academy of Medical Sciences. So this report was done in 2015 in collaboration with the BBSRC, the MRC and the Wellcome Trust. And they highlighted 6 causes of reliability problems. And I'll just quickly go over what these are so that we're aware of them. So omitting null results. So this is also known as publication bias. So when scientists or journals decide not to publish studies unless a statistically significant result is found, that can really bias our literature. Data dredging, so this is also known as p-hacking, and this involves repeatedly searching a data set or trying alternative analyses until a statistically significant result is found. Another problem is weak experimental design. So a study may have one or more methodological flaws, and this means that it's unlikely to ever produce reliable or valid results. Another issue is underpowered studies. So statistical power is the probability the analysis will detect an effect if it exists, but an underpowered study is too small to reliably indicate either way. So that means that all animals in an underpowered study are wasted. Another issue is errors. So technical errors may exist within a study, so a misidentified reagent or a computational error. And then the last one, which will be familiar from the previous slide, is underspecified methods. So even if a study is really robust, if its methods are not shared with other scientists in enough detail, then others can't replicate it. And I will be going over some resources later in my talk that can help us address many of these. So we know from the last couple of slides that there are not enough methodological details in publications to enable other researchers to reproduce experiments and build on them. But another thing that's really frequently missing from publications is information allowing readers to assess how reliable the results in a publication are likely to be. So this paper on this slide was published in 2020, and researchers used the software SciScore to analyze 1.6 million open access papers from PubMed Central. And the graph on the left is just looking at animal research papers published in 2018. And it shows that for these papers published in 2018, mentioning the use of measures to reduce subjective bias was really low. So randomization, that's our bar in the yellow, was only mentioned in 37% of the publications, blinding in only 12%, and power calculation in only 7% of publications. The graph on the right here shows the evolution of reporting these measures over the last 25 years across all of the papers they analysed. So these include preclinical and clinical research. So we can see that there is some improvement over the years, especially for randomization. So that is the second line down, the yellow line. That was about 10% of papers reported whether or not randomization was used in 2000. And by 2018, that was up to about 30%. But the prevalence of reporting these is still really low. Some groups have also broken this down by specific disease areas. So this is from the Camarades group in Edinburgh that Emily works with, and the graphs show the prevalence of reporting randomization, blinded assessment of outcome, and sample size calculation across different disease areas. So the disease areas in these graphs are listed in the bottom right of the slide, and top to bottom corresponds to left to right across the x-axis. So we can see that reporting does vary a bit for randomization and blinded assessment of outcome across the different disease areas. And on average, about a quarter of publications mentioned randomization, and just under a third mentioned blinding. But we can see that sample size calculation is really low across the board, so it's not mentioned across the board. So let's have a look at how these 3 things can affect the reliability of results. There are other things that we can do to help improve reliability of our research, but I'm just going to touch on these 3 things today because I believe the talk next week will help touch on things like systematically introducing heterogeneity to make our results more robust. So let's start with randomization. So these data are from a Camarades systematic review looking at the efficacy of interventions in vivo models of multiple sclerosis. So this study combined the results from 126 different studies testing the efficacy of 36 different drugs. And this graph is looking at studies that did use random allocation to groups versus studies that did not. On the y-axis here, we have the effect size. So that is the % improvement in neurobehavioral score for the animals given the drug. And we can see in the bar on the left, where animals were not randomly allocated to groups, the effect size was on average 42%. But in the bar on the right, where random allocation to groups was used, the effect size was on average 21%. So we can see that if you don't use randomization in your studies, then we tend to overestimate the treatment efficacy. So our results are obviously not as reliable. And this paper also looked at blinding. And so this is the same y-axis as in the previous slide. And the bar on the left is where blinded assessment of outcome was not used, and the effect size average was 41%. And the bar on the right, where blinding was used when assessing the outcome, the average effect size was 30%. So this shows that If you don't use randomization, you don't use blinding, you tend to overestimate the effect of your treatment, which obviously adds to reliability issues. But another problem that affects reliability of results is inadequate sample size. So this paper was published by Kate Button and colleagues a few years ago, and they were looking at sample sizes reported in the literature, and they found that many studies are badly underpowered, and the average power in neuroscience animal model studies was in fact between 20 and 30%. And this can be a real problem because low-powered studies really decrease the chance of detecting a true effect, but it also increases the chance that a statistically significant result you find is actually a false positive, and also tends to overestimate the effect size. So that's a problem on many fronts. But let's have a look at some resources that are available to help us improve the reliability of our results. So we've identified what the problems are, but here are some things we can do to fix it. So the NC3Rs have developed a number of resources to help scientists improve design of animal research and help improve its reporting in scientific publications. And NC3Rs resources are all available freely through the NC3Rs website. And I'm going to start with the resource, the Experimental Design Assistant, and this is to help you design studies more likely to yield reliable results. So the EDA is an online tool with a supporting website, and it's free to use, it's secure, and it was developed by experts in experimental design, statistics, and specialized IT systems. So the EDA has over 12,000 users, and it is recommended by major biomedical research funders such as the NIH and NSF in the USA, and the Wellcome Trust and the MRC in the UK. So you can find the EDA at eda.nc3rs.org.uk. And the EDA helps by providing you with bespoke advice and feedback on your experimental design as you plan your experiment. It gives you recommendations for statistical analysis methods that are appropriate for your specific design. It can help you determine the appropriate sample size through inbuilt power calculators, and it can help you with your randomization and blinding, including randomizing, taking account of any blocking factors or factors that you need to randomize separately. And when you create and design your experiment in the EDA asks you for a lot of information, which gives you a nice detailed experimental plan. And this can help improve the transparency of your experimental design. And that can really help you when you are sharing and discussing your plans in detail with colleagues and collaborators so that you can really talk it through and get on the same page before you start your experiments. And the EDA can also be used to help you teach experimental design. So in the EDA, each experimental plan is represented as a diagram made up of these colored shapes called nodes, and each of the nodes represents a part of your experiment. And there's more information inside each of these shapes that is specific to that part of the experiment. So for example, the group nodes, and those are the blue squares, they have the number of animals in that group, the number of experimental units in that group, because that's not always the same thing, and then a justification for the sample size. So you can put the details of your power calculation there, for example. And in the allocation node, which is the blue circle, we have information about how you're planning to randomize. So is that randomization within blocks or randomization within factors, but also information about how you're generating that randomization sequence. So for example, a spreadsheet generated by the EDA, or are you using another computer generated sequence or a Latin square? So you can be really explicit about what you're doing in an EDA diagram. You can also represent any experiment as a diagram, and the diagrams are machine readable. So the EDA uses computer-based logical reasoning to provide you with feedback. And feedback includes things like, for this experiment, it would include that the diagram doesn't include a nuisance variable. It goes on to explain that a nuisance variable indicates other sources of variability or conditions that may influence your outcome. It gives you examples of common nuisance variables in animal experiments, such as baseline body weight, and then how to use that variable in the design and analysis of the experiment. So it gives you different options, and you can figure out which one works best for your experiment and how they can help you mitigate against the effects of this variability in your results. So if you do want to use the EDA and you'd like a bit of help getting started, we are running live demonstrations regularly, and the next live demonstration is at the end of March. We also have a recording of a demonstration online, and both the live demonstration and the recording cover how to build a diagram in the EDA, how to get that feedback to help improve your experimental design, how to generate a randomization sequence, how to generate a PDF report that can summarize the key features of your experimental design, and of course, how to find the help that is built into the system. So another really important tool for increasing the reliability of our research are the ARRIVE guidelines. So the ARRIVE guidelines were developed to support researchers when writing manuscripts. So they're a checklist of key information to include in the scientific paper. And this is really key for several reasons. So it can help the research be evaluated adequately. It can help the methods and findings be reproduced. And they were originally published in 2010, and they've been updated to a new version that was published in summer 2020. And these are now split into 2 sets. So we have the Essential 10. So this is the basic minimum to include in your manuscript. So without this information, readers and reviewers can't assess the reliability of the findings you're presenting. And then we have the recommended set. And this really complements the Essential 10 and adds really important context to your study. So reporting items in both sets represents best practice. And the ARRIVE guidelines have also been updated to reflect current best practices. For example, there are now new items regarding protocol registration and data access. So to help understand why we ask you to report this information and how to and what to report, we've developed an explanation and elaboration document. And this is also available on the ARRIVE Guidelines website, which is arriveguidelines.org. And in this document, we've clarified some key concepts, especially those around experimental design and statistics. And we also provide examples from the published literature showing you how to report this information thoroughly. So that can help us improve reliability of our results and publications. Another way to help increase the reproducibility of your experiments is pre-registration. So pre-registration is standard practice for clinical trials, but we think that it can really help animal research. So we should be making it standard practice here as well. So there are different options to pre-register animal research. So you can register in specific animal research registries, but you can also directly pre-register your experiments with the journal where you want to publish your study. So that's a specific article type called registered reports. So in this case, you submit your protocol, that gets peer reviewed, and you can improve your experimental design before the experiment starts, and then you get a publication accepted in principle based on the protocol before you start collecting your data. So pre-registering your experiments with either of these methods is a sign of good rigor and reliability. So it shows that you've planned your research rigorously, but it also demonstrates your hypothesis, your approach, the analysis plans, that these were all determined in advance and were not shaped by your data as it emerges. So having a detailed record of your experimental design and analysis plans helps you differentiate between what the experiment really was specifically designed and powered to test and really exciting observations that you have noticed but need to be rigorously tested in a future experiment. And pre-registration also provides evidence that your results have not been selectively reported. So you're publishing all of your experimental groups and all of the outcomes that you measured. And there's another important initiative I'd like to highlight, and that is reproducibility. So reproducibility is a grassroots initiative helping researchers discuss issues, papers, ideas about open science, reproducibility, and the open science movement in general. And it is for researchers in any field and any career stage. And if you're interested in finding out more, their website is reproducibility.org, and they do have starter packs on the open science framework, and I've put the link on this slide here. And the resources on the OSF site include reading lists to help you get started, and there is one tailored specifically towards in vivo studies. So thank you very much for the whistle-stop tour of ways we can improve our reliability, and I'm happy to take any questions.
Wonderful. Thank you, Esther. That was indeed a whistle-stop tour, but I think incredibly informative. I particularly liked how you clearly set out the issues, but then also gave people some solutions, which I think is important, and I suspect why many people have joined us today, because they want to improve what they're doing in terms of their research. Another thing that I like as well is your focus on, before you start your experiment with the EDA, and how you report it. I think those are two clearly important aspects of our research. I might start with a question, but encourage people in the audience to take advantage of the fact that we have Esther with us this afternoon and her expertise to enter a question into the Q&A box. And I guess just focusing on that point that I'm making about the EDA, design and reporting, are there, or does the NC3, and maybe this is a very big question, but does the NC3R other aspects of the research pipeline that specific tools or strategies that they could implement would be useful to further strengthen in vivo research?
I think if you take things like you've got the EDA when you're designing your experiment, and the ARRIVE guidelines, actually the explanation and elaboration document has lots of good information about experimental design. So I recommend people read that when they're designing their experiments as well, especially because the ARRIVE guidelines were designed for writing your experiments, but as we found out in the reproducibility project slide, if you don't write down what you're doing as you do it, you won't be able to report it if you can't remember what it is. So I think ARRIVE and EDA can both be used to help you plan your experiments. And then we really recommend people pre-register their studies. So use something like preclinicaltrials.eu or the Animal Study Register. And then when you come to conducting your experiment, you could use the ARRIVE guidelines as kind of an aide-memoire to remind you of the things that you need to be recording and writing down, because you'll need to report them later. So you can kind of use that even while you're conducting your studies, just to remind you, okay, yes, I need to write all this down. And then obviously when you're writing your study, you've got the ARRIVE guidelines. If you've designed your experiment in the EDA, that actually collects most of the information from the ARRIVE Essential 10. So if you are writing your study and you've designed your experiment in the EDA, you've got a lot of that information. You've got your study design, you've got your justification for sample size, you've got how you used randomization and blinding, you've got all that information already. So that can really help you write up as well. And then I also find that when you're reviewing papers, I've started reviewing papers with the ARRIVE Essential 10 next to me, because that stops me from forgetting to check for something when I'm reviewing a publication. So I think we can use ARRIVE and EDA basically throughout the research cycle to help us, because I mean surgeons use checklists, pilots use checklists, even if we know we can forget things, especially if we're really busy. So having something like the ARRIVE Essential 10 to remind us, okay, yes, I need to mention this. And people omit some things not because they deliberately don't want to tell you, but they've just forgotten to mention it. So I think it really helps to have something just to remind us, oh yeah, I've got to include that, I've got to include that. So yeah, so I think you can use them throughout the research cycle, even though they were designed initially with a specific point in the cycle.
Thank you. We've got a few questions that have come in, so I will run through them. So one question, and I had a similar question actually, was do you have any idea how much the EDA is used by researchers? It seems like a no-brainer, I agree. But they're wondering how widely accepted it is or expected as a standard part of planning.
So
okay, I'll let you ask that first.
I was going to say we have kind of 12,500, just shy of 12,500 users registered for EDA accounts, but I'm very aware that the way you plan experiments is maybe you'll make a few experiments, design them, and then if the experiment takes 2 or 3 years to carry out, you won't be designing a new experiment for a few years. So I understand that it doesn't mean we've got 12,000 people making diagrams every week. So I've just calculated, actually, we have on average 54 new EDA diagrams created each week. So we know that it's being regularly used. And if we look at the Google Analytics of who visits our website, there's a nice little kind of curve for each weekday. So it goes down on the weekends and there's more use throughout each week. So we know that it is being regularly used. And there are, I think, 20 funders that recommend, if you're designing animal experiments, that you use the EDA to design your experiments. So they're suggesting this because it will remind you of things like, you haven't told us if you're blinding here. What is your randomizations? So it's reminding you of these things that will help make your experiments more reliable. And I think funders recognize that. And we have quite a few institutions and universities also recommend on their websites that people use the EDA if they're designing animal experiments. So we do have quite a few people who kind of endorse the use, but I don't think there's anyone who says you absolutely have to use it. Yeah, and I think it's also useful for ethical review applications as well. Like the PDF report that I mentioned really briefly in the talk is, it highlights the key information. So we actually worked with research funders to say, what's the key information you need about animal experiments? And the PDF report pulls that key information out of your EDA diagram. And so it lays it out in a really nice kind of easy to find format. So it's easy to see if there's any information missing. It's easy to see how many animals people are using, how they justified that sample size, and what they're going to do in their experiment. So I think it's actually quite useful also for things like ethical review, just so that people can review easily and a kind of standardized format, so you can easily find information for randomization and blinding rather than like weeding through lengthy paragraphs or something.
And I guess it's a bit of a time saver because it stops you having to repeat this. If you've got this one PDF, you need that information in lots of different contexts. Everyone who does research knows that you essentially have to write the same thing in different ways for different stakeholders. To be able to use having that information in a collated place that you can share more widely is, again, a no-brainer. Okay, we've only got 5 minutes and we've at least got 3 more questions in here.
Okay.
Are there tools, resources to assess experimental design of published papers? Poor reporting often makes it difficult to judge.
I have to say that for myself, I tend to use the Arrive Essential 10 and try and go through, because the Essential 10 is basically about how reliable, what are the key indicators that you need, or key information you need to know how reliable those results are likely to be. So I tend to go through the Essential 10 when I'm reading a paper, and key things that I look out for are, have they justified the sample size? And it doesn't have to be a power calculation. They could justify it with feasibility reasons, but have they tried to explain why that sample size was used? And the 3 main things I talked about today were the things I tend to look out for. Have they randomized? Did they random allocate to groups? And did they use randomization anywhere else in their study? For example, order of measurements or order of treatments? And did they use blinding at any stage? And if you are looking to increase the reliability of your results, just pick something easy that you can implement. So blinding may not always be possible throughout your experiment, but I don't actually know of a case where you can't blind in the analysis. So if you want to make baby steps towards improving reproducibility, start with blinding. Start with blinding your analysis. Then once you've got that embedded in your processes, then pick something else. So even if we make just little small incremental improvements over time, that will really improve the reliability of research. So say, if it all seems daunting to attack everything at once, then pick something that you can easily implement and start there.
For sure. But I think it is an important point that, Arrive is a reporting checklist rather than a assessment tool to judge the quality of reporting. And initially it might look like you could just map, one onto the other, but sometimes it's not quite as straightforward as that. So, there are some tools that exist in different contexts for in vivo research. There are a few, but then again, not widely used. So I think there's some work around that for sure.
Yeah, and I would reiterate Emily's point that yes, Arrive, we're about reporting, and it's like if it's about knowing what you've done. So it doesn't say you absolutely have to have done something, but you need to mention whether or not you've done it. And there may be a really good reason for not having included blinding in a certain stage of your experiment, but it's important that readers of your papers know that. So it's about telling us what you've done.
Yeah, communication for sure. Does the EDA work for exploratory and pilot experiments, or is it just for rigorous hypothesis testing?
So the EDA was originally designed looking at hypothesis testing experiments. So it is best at looking at hypothesis testing experiments, but you can use it for pilot studies or for exploratory studies, because the same kind of principles apply, right? The experimental design principles still apply. If you're doing an exploratory study, if you can use blinding, then that will make your results more reliable. So I think if it's an exploratory study, then maybe the statistical analysis suggestions may not be appropriate, because you may be looking at more outcome measures because you're still exploring which one would be the best one to answer your scientific question. So maybe some of the more statistical advice may not be as relevant, but it still could really help you communicate your experimental plans with others. And the more rigorous you can make your exploratory study, the more reliable those results are going to be. So the more it can help you contribute towards perhaps a future hypothesis testing study.
For sure. We've only got 1 minute left. Alina Christina has asked for resources to help with doing power calculations. I'm hoping that they can be shared afterwards if it's just specific resources. But the question, I think that it's a question that I'm asked the most, so I'd like to get your thoughts on this, is the case for pre-registration of animal research looks really strong. But this person, they don't do it in their lab, and their supervisor feels it's impossible to do on a practical level. Do you have any thoughts on this?
So I know there are some people that have contacted me about pre-registering more exploratory experiments, and they've said, like, what do I do in that case? And you could pre-register a sort of decision tree type of experiment. So if what your first experiment gives you determines what you're going to do in the second experiment. You could pre-register, okay, this is what I'm going to do, and if I get this, I'll do that. If I get B, I'll do this, and if I get something completely different, then I'll sit down and replan. So you can, that is one option, to pre-register an if-then type scenario, because I know that, pre-registering was obviously initially designed for clinical trials where there's very defined outcomes and randomized control trials where it's very structured how they're carried out. But I think we can still adapt pre-registration for more exploratory work, and it can still be really useful.
And I think part of it is a cultural thing. It's not the cultural standard in vivo research to pre-register. As far as I'm aware, I could be wrong, there aren't any registered reports of in vivo research. So I think we're still on that journey into normalizing that. Okay. I think I'm going to wrap up there. It does feel really short, doesn't it? There's so much more we could talk about. I felt like I had to cut you off in certain bits there to get through it all, but I'm glad we managed to get through the questions that were asked. So I want to say a big thank you to you, Esther, for taking the time to put together your slides and share your knowledge with everyone. I want to thank the audience for being with us this afternoon. Remind them that this is the first of the 3 in vivo webinars that we're hosting. Next week, we have Natasha Karp from AstraZeneca, who will be talking about embracing variation to improve reproducibility. So I hope that you're able to join us next week. And I think with that, I will let people go. Thanks again, Esther, and thanks to everyone for joining us. And I hopefully will see you next week.